Hi everyone, my name is Matt Rogers and I'm a second year chemical engineering graduate student working in the Zanic Lab, and we're trying to understand the dynamic architecture of the microtubule cytoskeleton. So I know since you guys can't be here with us on campus, I just wanted to throw in a quick little Google Earth image of the campus just to show you a little bit of what it looks like, better than nothing. So in order to understand what our lab does, we first need to introduce microtubules. And microtubules are complex biological polymers. This is a snippet from Harvard University's Inner Life of the Cell video, where we have tubulin dimers polymerizing into a hollow cylindrical lattice that is a microtubule. And this lattice can polymerize and it can depolymerize, as we're seeing now, rather spontaneously. And microtubules are used in a variety of cellular processes, including vesicle transport. And this is really cool. It's really cool to see these little kinesin molecules walking along a microtubule, and it's just really cool. But they're used in a variety of cellular processes. So as I mentioned, microtubules are highly dynamic, as we can see in these videos that show microtubules in red, and the green helps show the edge of the microtubule growing and shrinking spontaneously over time. And they form this dense cytoskeletal architecture or network going throughout the whole cell. So why do we care about microtubules in general? I mean, what's the high level overview? What's the goal that we're working towards? And so we want to understand microtubules better because they're implicated in a variety of diseases. So cancer is characterized by uncontrolled growth and division of mutated cells, and microtubules are critical to mitosis or cell division. And a lot of cancer therapeutics actually target microtubules specifically. Microtubules are also implicated in Alzheimer's disease and heart failure. So there's a wide array of maladies that better understanding microtubules could help us treat. So just to briefly walk you through a typical day in our lab and the different techniques that we use to help us model microtubules. So the first thing that we typically do when running an experiment in the lab is create stable nucleation sites, and we call them seeds. And so what we do is take a form of tubulin, put it in a solution, incubate it for a certain amount of time, and over that time, the tubulin polymerizes. Now, this tubulin is special in that it doesn't spontaneously depolymerize like we just saw in the cells. Rather, these seeds are meant to serve as templates or nucleation sites for the dynamic tubulin to polymerize and depolymerize as it would. And so we take this array of polymerized and unpolymerized modified tubulin, spin it down in a centrifuge, get stratification of the polymerized polymer from the unpolymerized monomer, discard the supernatant, and we're left with just the seeds or just the templates from which we can create actual dynamic microtubules. Then we move on to turf microscopy, and our turf microscope is shown here. And turf stands for total internal reflection fluorescence. It's a really cool microscopy technique where you actually excite a laser. The laser bounces off the glass slide in your sample but creates this evanescent wavefront that penetrates a couple hundred nanometers into your sample, and so you actually get a very thin slice of your sample that you can focus in on and see with really high resolution. And so you don't have a lot of the above and below noise that cancels out your signal like you would in confocal microscopy, for example. So this is an image of actually growing microtubules. Those are the green extensions that are growing and shrinking over time, and they're growing from this template, this seed microtubule. And so you can see over time they grow, they shrink. We see this dynamic instability. And so we actually use a variety of chemical engineering techniques in this study, including reaction theory, thermodynamics, looking at equilibrium constants, free energy given off as a result of polymerizing or depolymerizing. And so we actually can use several different analysis techniques to help us understand different microtubule properties. And one of these techniques is Matlab modeling. So we can take these differential equations that describe microtubule mechanics, plug them into Matlab, and we can see that the predictions made by the Matlab model are pretty well in line with the actual microtubule results that we see when running these experiments. Furthermore, we can use COMSOL, a computational fluid mechanics modeling software in combination with microfluidics in order to yield very robust flow profiles in the device and help us better understand microtubule mechanics as well as their dynamics. And we can actually take it a step further and even model the dynamics themselves using computational modeling. And so this is a Matlab prediction of a microtubule growing over time. And so we're looking at the edge of this microtubule that we just made green because that's the fluorescent color we use, and compared that to what we see actually in our experiments. And you can see that we have very good agreement between the actual condition that we see in vitro and the prediction made in silico, which is really cool in my opinion. So our lab isn't all work, no play. We have a really good lab dynamic structure. We do things like birthdays. Everybody gets a birthday cake when it's their birthday. We take hikes, go on kayaking trips. This is pre-coronavirus. But we spent a lot of time outside the lab together, hanging out, doing Zumba classes, that kind of thing. It's a really great dynamic. So please feel free to reach out for more information or to have any of your questions answered. My email is on this slide, as well as the PI Maria Zanic's email. Would be happy to talk with you. And just want to thank you guys for your time, and hopefully we'll see and talk to you soon. But have a good day, y'all.